Welcome to Strand NGS. This tutorial will help you in getting started with pathway analysis. Strand NGS supports pathway analysis on well-studied curated pathways and literature-derived networks. To begin pathway analysis, you will need organism-specific pathways and supporting databases. Now we will walk you through the steps to import the curated pathways, literature-derived networks, and download supporting databases. Firstly, you will need to update supporting databases, that is Interaction Database, bridgeDB, and Homologene. Go to Annotations Annotation Manager, click on the list. All the organisms and their associated annotations that are available in the Strand NGS server are listed in this panel. Expand the organism, check the box corresponding to bridgeDB Interaction DB Homologene groups and click on Update. Once the update is completed, close the Annotation Manager window. Organism-specific pathways available at wikipathway portal can be imported into Strand NGS via the main menu. Go to Annotation, select Import Pathways from wikipathways. In the Choose Organism dialog, to import pathways for all organisms, choose All Organisms, else choose Select Organism of Choice from the drop-down menu and click on OK to import the respective pathways. If you have a pathway file in BioPax Level 2, BioPax Level 3, TXT, or GPML format, you can import the same via option Annotation Import Pathways from File. Choose the options as per your file format and proceed with the wizard. Now we will brief the options present in Strand NGS through which pathway analysis can be performed. Option 1. The Single Experiment Analysis, or SEA. SEA identifies matching pathways for one experiment. If SEA is chosen, by default active experiment is selected for analysis. In step 1 of 4, select the pathway organism from the drop-down corresponding to choose pathway organism. The list of organisms available in the drop-down depends on the pathway organism for which you have previously downloaded pathways or the interaction database available from the Strand NGS server. The organism in the experiment can be different from the pathway organism. If the pathway organism is different from that of the organism selected in the experiment, the software looks for homologene translation and bridgeDB ID to map matching entities from different organisms and different annotations. If the pathway and experiment organisms are the same, but the bridgeDB for the organism is not available, then the workflow proceeds further but only identifies direct matches based on available identifiers. In step 2, specify interpretation, entity list, and annotations. The input entity list and the pathways from the selected organism are matched based on these annotations identifiers. In step 3 you can review the results. The pathway column provides the name of the pathways present in the experiment. P-value column represents p-value of each pathway entity. The Matched Entities column contains the number of entities in the pathway that matched with the input entity list. Pathway Entities of Experiment Type column contain the number of entities in the pathway that are of the experiment type. Sort the columns, and you will be able to see the maximum number of entities in the pathway that matched with the input entity list. If you would like to save only entities in the pathway that matched with the input entity list, then select respective rows and choose Custom Save List, on Next and Finish. The result will be saved in Project Navigator. Click on the full screen for better visualization. The pathway result view comprises of 5 sections. Each one of them is dependent on others. This section gives information on the interpretation and entity list that was chosen to perform pathway analysis. This is the pathway list panel, information on pathways for the chosen organism, p-value and a minimum number of matches is available. Here is the filter panel, you can filter pathway lists by p-value cutoff or the minimum number of matches for each input entity list in order to identify significant pathways. Schematic representation of a pathway is displayed in the pathway viewer. Double-click on each pathway and the pathway will be displayed in the pathway viewer. This is the heat map pane. All the pathway entities of the experiment type in the pathway entity column are listed here. For better visualization of the pathway view, click on Zoom In icon in the pathway view toolbar. Matching entities between the pathway and selected entity list for C are marked in yellow color fill. All the entities in the pathway can be selected with this icon. This icon allows you to select matched entities in a pathway viewer. The matched entities are marked in yellow color fill. You can select them by click on Select Highlighted Entities icon. This icon enables you to create an entity list that is selected in the pathway viewer. Selected entities can be visualized in the profile plot. Another entity list and interpretation can be chosen with this icon. Quilt plots or heat strips can be displayed with this icon. Since I have created SEA with the non-averaged interpretation, you will be able to see only quilt plots in the pathway viewer. These are the quilt plots for each sample. My dataset contains 6 samples, hence you are able to view 6 colored blocks representing the expression levels in each of the 6 samples. This icon will help you in changing the display settings of the quilt plots. To display quilt plots or heat strips or remove the quilt plots, you can use this icon. Each matched entity is highlighted by borders. If you wish to make changes with respect to the border, choose this option. To modify the display settings such as color and experimental data values, you can choose this icon. The icons in gray are not applicable to this specific analysis, hence they are grayed out. Option 2. Multiomics Analysis or MOA. MOA is used to identify matching pathways between 2 experiments, and these experiments should be present in the same project. If the experiment is present in a different project, it can be added to the existing project by clicking on Add Experiment icon present in the menu bar. Through MOA workflow, it is possible to visualize overlapping entities of same or different analysis type. Choose 2 experiments that you would like to compare and click on Create MOA. Proceed with Pathway Analysis workflow. In Step 1, you will see information on the 2 experiments and the associated organisms. Select Input Entity Lists, Interpretation, and Annotations for both Experiment 1 and Experiment 2 and click on Next. At this step, you will view the p-values, number of matched entities between the input entity list, and the pathway and pathway entities of experiment type are listed in their respective columns for each input entity list. You can either custom save or click on Next and finish the wizard. The sections in the pathway result viewer is similar to that of SEA, but you will view 2 sets of p-values and number of matched entities columns. Option 3. NLP Network Discovery, Natural Language Processing, NLP, algorithm runs on the published Medline abstracts. NLP is based on a deep parsing method and operates based on sentence-by-sentence manner and extracts only those relations that are completely within a sentence. Choose the entity list of your choice. The simple workflow includes 9 predefined algorithms, and these options help us to determine the type of analysis performed. The advanced workflow consists of 3 predefined algorithms. Each one is good enough in their own way. To get more information on this, you can click on Help, go through the manual, and choose the appropriate algorithm as per your requirement. Direct Interactions finds relations that connect the selected entities. I will go ahead with this option itself. At this step, you will find initial number of entities, that is the number of entities in the input entity list. The number of new relations and the number of new entities are also displayed. This icon enables you to view entities in different layouts. I will launch the cellular view just to showcase how it looks. This is an example to represent directly connected entities. These are the unlinked entities. Unlinked entities can be removed with the help of this icon. A new entity can be added with this icon. A new relationship can be added with this icon. This is the entity legend. Each shape gives an idea about the role of each entity in the pathway viewer. Relations legend gives an understanding of the types of relationships between the entities. Edges legend depicts relations. Click on Next and finish the wizard. Option 4. Mesh Network Builder. MESH is an acronym for Medical Subject Heading and runs on Woodline abstracts. It is a novel way of creating pathways around a term or a concept. Strand NGS pulls out the mesh terms relevant to the input term, concept from the mesh database, by exact, substring, synonym matching. Type the concept or the mesh term here. Since I am working with Alzheimer's sample, I will enter the mesh term as Alzheimer's disease. This table lists all the mesh headings containing the input term Alzheimer's disease. If you would like to include only those interactions that contain the exact mesh headings that were selected, then choose Exact Relations. If you would like to include all interactions that contain either the exact mesh heading or the child mesh terms, then choose All Relevant Relations. Minimum frequency defines the minimum number of PubMed articles, MIDs, associated with the mesh term that interactions should have. The default setting for this parameter is 1, you can change it as per your requirement. Select the mesh heading, define the filtering options and move to the next step. The pathway built out of all the interactions associated with the chosen mesh term or terms is shown here. In this step all the details about the creation of the new network are summarized. Click on Finish to create a pathway. Option 5. Extract relations via NLP. It is a mechanism to create new pathways in Strand NGS. If you would like to create a pathway on the inbuilt NLP algorithm directly on PubMed abstracts, then choose PubMed Search. Choose File to run NLP on local PDF, doc, or HTML files. NLP first recognizes entities and sentences and then performs information extraction to identify relationships between these entities. Specify the query here. Aricept is one of the medicines used to treat cognitive symptoms of Alzheimer's disease, hence I will enter this term here. The PubMed ID is shown in the left-hand column. Each tagged document can be viewed separately in the right-hand column by selecting the corresponding document name or PubMed ID. The target documents containing the search terms are identified and tagging is performed using the entity dictionary. All molecular and biological process, function entities present in the Strand NGS interaction databases are tagged in these searched documents. Matching entities are highlighted according to default color settings. All sentences which have at least 2 tagged entities are analyzed by the NLP algorithm. A pathway view is launched with the relations extracted from these sentences. The total number of extracted relations and new entities is also shown here. The sentences from which the relations were derived can be examined by double-clicking on the relations. Click on Next and finish the process. The literature-derived pathways can be used to perform SEA and MOA. For this you will need to choose the literature-derived option here. The features and capabilities of pathway analysis in Strand NGS are enormous. We have briefed about the basic concepts involved. You can explore the pathway analysis feature at any point. If you have any question, write to us at support@strands.com and we'll be happy to help you. Thank you for watching the tutorial.